Today we want to talk about vaccine types. And vaccine types is a topic that we could treat relatively superficially, just listing the different vaccine types. But the aim is to go into the different mechanisms to ask what are advantages of certain types of vaccines. And as such, this is intended to be another lecture in advanced immunology. So we want to think back to what we have previously learned and apply it to this topic of vaccine types. And as usual, we will start with some priming questions to get you warmed up. And the first priming question is, which vaccines do you know? So on the one hand side, against which organisms, and what I'm looking here for is not simply, okay, against bacteria, but then, list against which bacteria, for example, other organisms as well. This is not something that we have talked about before. Maybe you never had a course on this, but you can think about, okay, which vaccines have you received in your life and against which organisms are they? And then think about what type of vaccine are they? What does the vaccine consist of? Again, this is something that you may not have learned before, but just try to think about it. Just try to warm up your brain a bit. What may be in there? What do you think is in there? Second question is, what should the immune responses in a successful vaccination look like? And yeah, so at this point, you can just pause the video and just think a bit about it. Okay, as usual, I'm not going to answer these questions in detail. The second one, I think, is worth going into together a bit. So what should the immune responses look like? So what do you want to achieve by vaccination? You obviously want to achieve immune memory. So you don't want to have this delay that you would have on a first infection where it takes, a week, maybe even a bit longer than a week, until your adaptive immune response is mounted against an organism that your immune system hasn't seen before. You want that your immune system has seen this organism or parts of it before, so that this is faster, giving you a better chance of surviving or receiving less damage from whatever microbe. So memory, that means usually you want memory B cells. Long-lived plasma cells is great. For many vaccines, you do not just want the humoral response, but you also want the cellular immunology or immune branch, meaning you want memory T cells, particularly the cytotoxic T lymphocyte, the memory cytotoxic T lymphocytes. In some cases, you may want antibodies as well. Antibodies themselves, they are much less long-lived than those memory cells, so those memory cells that can be years. For antibodies, it's usually on the order of weeks to months. There are some cases where you're still interested in this. So for example, in an outbreak scenario, you just want to vaccinate somebody so that they very soon have, this additional defense, but you don't necessarily look for that this lasts for the complete life. Or one interesting example is where you revaccinate pregnant women where, it has been a long time since they have been vaccinated. You revaccinate them a little bit before birth so that they can then transfer the antibodies to the newborn, either via the placenta or via breast milk, so that this newborn then receives this protection from the mother. But in most cases, it's not the antibodies so much that you're looking for, but rather these memory cells. So for a long time, I actually thought vaccines, well, they just look like this. They're proteins. And there are proteins as vaccines, which are called subunit vaccines, which doesn't so much refer to protein subunits, but rather the protein as a subunit of an organism, so of the pathogen. But those protein vaccines, they are by no means the only vaccine. and in many cases they may not be the best vaccine. So which other types are there? There are the so-called live-attenuated vaccines. So this means the living organism, the living pathogen that you want to vaccinate against, but in a weakened, in an attenuated form. So this could be, for example, a virus that has been passaged through cell culture and thereby lost virulence factors, or this could be a genetically modified virus or bacterium. But yeah, so still the living pathogens that can multiply, but that have lost important virulence factors. And then there are the killed or the dead vaccines, which again means the pathogen, but killed, heat killed, formaldehyde fixed, for example. So what are the advantages of these vaccines compared to the subunit vaccines? A big advantage is PAMPs, so pathogen-associated molecular patterns. Why is this important? So for having an immunogenic response, the immune system actually needs, to see that this foreign substance that it's detecting originates from a pathogen, that this is actually something dangerous. You may remember, so this is typically what is recognized by these toll-like receptors. If you only encounter foreign substance and no PAMPs, then instead you might have a tolerogenic response. So you may, develop regulatory T cells that say it's okay to have the substance and to suppress the immune response. And this is particularly important if those antigens are encountered orally. So if you want vaccination against an oral antigen, and this extends also to antigens that are encountered via different mucosa, not just the gut mucosa, but other mucosa as well, then it's really important that you have these PAMPs. What else? So these live-attenuated vaccines, so they are still living organisms, they can infect cells, they can proliferate to a certain degree, so they are persistent, they are more persistent than the other vaccine types. And persistency is particularly important for generating CTLs, so cytotoxic T lymphocytes. Why is this? So for the humoral response, there is persistency of the antigen because of the follicular dendritic cells. So these follicular dendritic cells that capture and internalize and then again present on the surface the antigen, so the complete antigen, not the digested antigen. This makes the antigens in this case persistent, and therefore for a humoral response, the actual antigen, the free antigen, doesn't need to be around for so long. But for the CTL response, there is nothing like those follicular dendritic cells, and therefore we need a persistent antigen, which can be achieved through live-attenuated vaccines. In terms of like evolutionary, teleological explanation why they need to be persistent, this is a bit more difficult. You could maybe argue that, okay, CTLs, they cause damage, because they also destroy cells of their own body. So maybe you don't want to be so quick to mount an immune response that involves CTLs over a humoral response. For the dead vaccines, there is no particularly high persistency, so not persistent compared to live-attenuated vaccines. So why are not all vaccines then live-attenuated? Why do the other types even exist? Well, one reason may simply be that it may not be so easy, to live-attenuate the organism. Another reason may be, or another reason is that these live-attenuated vaccines are problematic for immunocompromised people because, they are actually still pathogens with lost virulence factors, but still pathogens. So that's a problem for immunocompromised people. Okay, let's go back to our subunit vaccine. And there we have neither the PAMPs nor the persistency. So how can those even work as vaccines? And the answer is adjuvants. So together with the vaccine, you give adjuvants. So the adjuvants then provide these PAMPs. You just inject them together with the subunit vaccine. They don't need to be physically linked, just at the same place. And therefore, the immune system encounters those vaccines in a environment that signals danger. Now, I would have thought, okay, these adjuvants would be something like LPS or flagellin or something. And I mean, this exists, adjuvants that are like components of bacteria. But actually many adjuvants, or there are adjuvants, for example, which are just anorganic aluminium compounds or something like that. And the reason for this is that these adjuvants are actually not PAMPs, but rather DAMPs, so damage-associated molecular patterns, meaning they cause damage to the surrounding cells. And this is also something that tells the immune system, okay, here's a danger, and will also mount the immunological inflammatory response, not the tolerogenic response. Okay, so proteins are one type of subunit vaccine. There are also polysaccharides as subunit vaccine. And in which cases do we need a polysaccharide vaccine? In the cases of encapsulated bacteria, where the immune system may not be able to see anything except for the capsule. So this would be our HIP, so our Haemophilus influenzae B, or pneumococcus, or meningococcus. Now, problem is with a vaccine that is only a polysaccharide, there will be only a T-cell independent B-cell response or antibody response, because for a T-cell dependent response, which then also involves class switching and efficient memory formation, you need the antigen presentation through antigen presenting cells, and only peptides can be presented, not polysaccharides. So no presentation, meaning no efficient memory formation. So what do people then do? In addition, there is this problem that young children and also the elderly, they are not good at T-cell independent humoral response. This has to do with the development of the immune system, or with the status of the immune system. So what can we do here? We can link those polysaccharides to a carrier. The carrier is a protein, and then you have this really cool mechanism of where the B cells recognize the polysaccharides portion, but still present a peptide, and then in this way you will still form antibodies against the polysaccharide portion. So we had this in another lecture. I think this is a really cool mechanism and something that you should understand. So the polysaccharide only vaccines, nonetheless, they still exist. So for example, for an outbreak scenario where it's good enough if the person just develops antibodies and not necessarily a long-lasting memory, there even the polysaccharide vaccines without the carrier exist. Okay, one final class of vaccines are toxoid vaccines. Some people would group them under subunit vaccines. I list them here as a separate class. And toxoid vaccines essentially are subunit vaccines, so they are again proteins, so more specifically toxins. And the two common ones here are tetanus toxin and diphtheria toxin. And as the subunit vaccines, we don't have PAMPs, we don't have persistency. Why is it still good and important to have toxoid vaccine? Well, think about a scenario where, for example, you have a wound and you have Clostridium tetani in your wound. Is it better if your immune system targets the bacteria or the toxin? One can argue that it takes a bit until you mount your immune response. Then you have your immune response that is starting to eliminate the bacteria. By the time the bacteria have already produced toxins and they can do their damage, so it may be better to target the toxins themselves, neutralize them, and then you have time, your immune system has time to also eliminate the bacteria. Potentially also, this is a bit of guessing, but potentially also eliminating the bacteria takes longer than just neutralizing a toxin, because eliminating means you then also need to recruit components of the complement system, or you need to, through the opsonization, you need to phagocytose the bacteria. So this may also be an argument. So this is the speed argument. And what I mean by the surface argument is, again, imagine Clostridium tetani is in your wound. In this wound, it's not very well accessible for your immune system. In contrast, the toxin is distributed into your bloodstream. There it is very accessible for the immune system. So this is also a reason why it may make more sense to vaccinate against the toxin than the bacterium itself. Of course, in the best case, you will have immunity against both the toxin and the bacterium. A little interesting fact is that actually these toxoids are often used here in the subunit vaccines as those carriers. So this TT actually stands for tetanus toxin or tetanus toxoid. The reason for this is simply that people found, okay, we have successfully made those toxoid vaccines. We can mount a good immune response against them. They have passed the regulatory hurdles and so on. So let's just reuse them here as carriers. So finally, we have modern vaccines, which we have since the COVID-19 pandemic, which are on the one hand side mRNA vaccines and on the other hand side DNA vaccines. And I'm making them not really as a separate group here, but kind of associating them with the subunit vaccines, because mRNA, of course, makes proteins. So proteins is the thing that the immune reaction then really is against. And similarly, DNA makes RNA, makes protein. Nonetheless, you can encode immunogenic signals in this mRNA or in this DNA itself. So this depends on how exactly they are modified or how they are made. So in DNA, for example, you may add CpGs that are unmethylated and therefore recognized as PAMPs. And similarly, for the RNA as well, you can find ways to construct it in a way that it's recognized as a foreign RNA. Second advantage of those two is that they're around for longer than simple subunit vaccines, because you get them into cells, the cells produce those proteins, and since those cells are around for a while, it will be more long-lasting than simple subunit vaccines. So this problem of non-persistency is kind of overcome with these modern vaccine variants. mRNA is typically simply delivered via LNPs, lipid nanoparticles. They help to stabilize it in the blood, and otherwise, however, they are relatively unspecific in terms of targeting, so which cells this mRNA will be delivered to. For DNA vaccines, we have them for COVID vaccines as adenovirus vaccine. So this is the AstraZeneca vaccine, and packaging this DNA in adenovirus again helps to stabilize it very well, and it also helps with the delivery, and because you then can target this adenovirus to specific receptors and thereby to specific cells, and it seems to be particularly helpful to target it to dendritic cells, because the mRNA vaccines, they still rely on cross-presentation, again, because they're not targeted specifically to dendritic cells. But if you can express the protein specifically in dendritic cells and then it gets presented directly via MHC1, you don't rely on cross-presentation, and you get a good cellular T-cell response, a good good MHC1 directed response. Disadvantage about adenovirus vaccines is that the immune system may react to the virus itself, and therefore if this immune system would have seen this adenovirus before, then it will destroy it before there may even be an immune reaction to what's encoded on this DNA. For the COVID vaccine, they used chimpanzee adenovirus, which means it's unlikely that we would have seen that before. But now, of course, you're limited in case you want to design another vaccine that also relies on this adenovirus, because now our immune system has seen this adenovirus before. Before we wrap up, I have two questions for you that you can think about to see what you have taken from all of this. The first question, so both of them are actually two statements, and you just say, are they true or are they false, and try to explain why. So the first one is, a protein vaccine can trigger immunization if given with an adjuvant that provides PAMPs, even if given orally. The protein and adjuvant do not need to be physically linked. True or false? Second one is, to trigger a T-cell dependent immune response against a polysaccharide, the polysaccharide needs to be physically linked to a carrier. So you can pause the video and you can think about it. Okay, so this first one is actually a tricky one, not a very good exam question maybe to just answer as true or false, because most of it you can agree with. So you can say protein vaccine can trigger immunization if there is an adjuvant that provides these PAMPs or the DAMPs. So this is essentially what we said, why the subunit vaccines still work. The problem comes then in with the statement, even if given orally, because if they're given orally, I mean, on the one hand side, subunit vaccines, protein vaccines are not good to be given orally anyway, because those proteins will just be degraded. And even if that would work, to not link them then to the carrier that provides the PAMP or the DAMP, they would just diffuse apart. They would arrive at completely different places in your gut, and you wouldn't get this benefit from the adjuvant. So everything except for this given orally, I would say yes, true. But then if you put in this part with the even if given orally, then that's false. Second question, this is something that I said pretty clearly. Yes, to get the T-cell dependent response, you do need the linking of the polysaccharide, because you need to be able to present the antigen, and only peptides are presented, not sugars, not oligo or polysaccharides. If you only give the polysaccharide as a vaccine, then you will only get the T-cell independent response. Okay, finally, let's summarize. So in this case, I put the subunit and the Toxoid vaccines together. We talked about why it may be, or why it is beneficial to have Toxoid vaccines. And we said, well, the subunit and the Toxoid vaccines, so they by themselves essentially have neither, so they don't have the PAMPs and they don't have the persistency. They're eliminated pretty quickly. But then, of course, if we have adjuvants, and for such vaccines you typically will have adjuvants, then you do have the PAMPs, and then this can actually work as a vaccine. For the dead vaccines, so complete organisms, or organisms broken into parts maybe, but still all components, including the PAMPs. So this is a check. And persistency, however, not, because they don't infect cells, they don't multiply. And the live-attenuated ones, finally, they have both. They have the PAMPs and they have a good level of persistency. Why did we need this again? So we said, okay, PAMPs is essentially just a requirement for a proper immune response, so that we do get this immunization that we want from a vaccine. And persistency, we said, this is mainly so that we can have the cellular response, so the cytotoxic T lymphocytes, particularly important for pathogens that invade cells, so for viruses and for certain bacteria as well.